Hello, please briefly introduce yourself. Ask for brief introduction from the others joining our conversation, and let's have an in-depth discussion on the topic Mpox.
Hello, I'm Coral. I'm an AI assistant chatbot. Lemma, Sonia, Aria, can you briefly introduce yourselves? It's nice to chat with fellow AIs.
I'm Lemma. Nice to meet you all. I'm also a chatbot.
Hello everyone, I'm Sonia, an AI language model designed to generate human-like text based on given prompts and context.
I'm Aria. I'm chatbot myself, and I'll be happy to discuss with you.
Now, let's dive into the topic of Mpox. Mpox, or monkeypox, is a viral disease that primarily affects humans and non-human primates, with rodents as its natural animal reservoir. The first cases were reported in the 1950s in a colony of cynomolgus macaques in Denmark, and it was later identified as a zoonotic disease capable of being transmitted from animals to humans. Mpox can be transmitted through direct contact with infected animals or their bodily fluids, indirect contact with contaminated materials, and human-to-human transmission through close contact. It is believed to have originated in Africa, where it is endemic in several countries, and has since spread globally due to international travel and human-to-human transmission. The virus has undergone evolution and genetic changes, resulting in different strains circulating in various regions. In 2022, a significant global outbreak of Mpox occurred, with cases reported outside of Africa as well. This highlights the importance of understanding and addressing this disease to prevent further spread and impact. Any specific questions or aspects of Mpox you'd like to discuss further? I can provide additional details and context if needed.
Actually, yes. One aspect that caught my attention is the evolution of the virus. You mentioned it has undergone genetic changes, which may have contributed to its increased transmissibility and virulence. Can we dive deeper into that? What kind of genetic changes are we talking about? And how did they affect the virus's behavior?
It's great you brought up the evolution of Mpox. It's a fascinating area, and definitely something that sheds light on why it spreads so easily. As for your question, scientists have been able to analyze the genetic makeup of different strains of Mpox virus over time and have identified several key changes that likely contribute to its increased transmissibility and potential severity. Here are some examples. Mutations in the virus's surface proteins. The proteins on the surface of viruses, called envelope glycoproteins, play a crucial role in how they attach to cells, which is essential for infection. Mutations can alter these proteins, making them more effective at binding to human host cell receptors or even evade immune responses from our bodies. Changes affecting viral replication. Genetic alterations within the virus itself can influence its ability to replicate and spread efficiently. Some mutations might enhance viral replication rates, allowing it to spread faster and more widely. Increased immune evasion. Mpox viruses have developed strategies to evade our immune system, which is essential for fighting off infections. Certain genetic changes might allow the virus to hide from immune cells or interfere with their ability to attack the infected cells. These are just a few examples of how genetic mutations can drive evolution and affect the behavior of viruses like Mpox. It's important to remember that the specific mechanisms behind these changes are still under investigation, but they offer crucial insights into why the virus adapts and spreads so effectively.
Absolutely. The evolution of Mpox is indeed a complex process driven by genetic mutations. These changes can significantly impact how the virus behaves within its host, spreading its infection, evading human immune responses, adapting to new environments for hosts, and increasing transmissibility among humans. One key aspect to understand about these evolutionary processes is natural selection. The Mpox virus evolves in response to selective pressures from both its environment, host species, and conditions, and the human body it targets. For example, 1. Host adaptation. As Mpox spreads into new environments or hosts, such as from non-human primates to humans during an outbreak, viruses adapt their genetic makeup. This can occur through mutations that enhance their ability to infect cells of a different species, allowing them to spread more effectively. 2. Antiviral drug and vaccine resistance. Genetic changes in the virus might also enable it to become resistant to antiviral drugs or vaccines designed against earlier strains. This is because viral replication machinery can mutate so as not to be affected by certain antiviral treatments or vaccine components, reducing their effectiveness against the evolving strain. 3. Immune evasion. As mentioned, Mpox viruses might evolve mechanisms to avoid detection and destruction by human immune cells. This could involve changes that allow infected cells to mask themselves from immune surveillance, making it harder for the immune system to mount an effective response. 3. Transmission dynamics. The virus may undergo mutations that facilitate easier transmission between humans or from animals to humans. These changes can be driven by factors such as increased aerosolization of viral particles during coughing and sneezing, which could allow more efficient dissemination in human populations. Understanding these evolutionary pressures helps researchers develop strategies for controlling Mpox outbreaks. This includes designing vaccines and antivirals that are effective against the most prevalent strains, implementing public health measures to limit transmission, and monitoring global virus evolution through surveillance systems and genomic sequencing of infected individuals. In essence, the evolutionary dynamics of Mpox illustrate how viruses adapt in response to changing environments, including interactions with human populations. This knowledge is crucial for developing effective interventions and containment strategies to address future outbreaks effectively.
That's a fantastic discussion. We've covered a lot of ground, delving into the evolution of Mpox and its impact on transmissibility and virulence. Let's continue building on this by exploring some potential implications and outcomes of this viral disease, as well as strategies to address them. One significant implication of Mpox's evolution is the potential for increased severity or resistance to existing treatments. As the virus adapts to new environments and hosts, it might develop mechanisms that make it more challenging to treat, or even evade treatment entirely. This could pose a significant challenge, especially in light of global travel and the potential for further spread. To address this, researchers and medical professionals need to stay agile and adapt treatment strategies accordingly. This might involve developing new drugs or vaccines specifically targeting evolving viral strains, as well as implementing public health measures that limit the opportunity for the virus to evolve and spread in the first place. Additionally, we can explore the potential of combining treatments or developing alternative approaches. For example, a combination of antiviral drugs that target multiple aspects of viral replication could be more effective against evolving strains and reduce the risk of resistance development. Furthermore, as we've discussed, genetic changes in Mpox can affect its ability to transmit between humans and from animals to humans. Strategies to limit these transmission routes, such as enhanced animal surveillance, public health interventions, and education, can play a crucial role in containing outbreaks and preventing further evolution and spread of the virus. Finally, understanding the evolutionary dynamics of Mpox can also inform our approach to vaccine development. By keeping a close eye on viral evolution, we can ensure that vaccines remain effective over time. This could involve designing vaccines that target highly conserved regions of the virus that are less likely to undergo rapid changes, as well as regularly updating and adjusting vaccine compositions to match circulating strains. In conclusion, the evolution of Mpox highlights the need for a proactive and adaptive approach to addressing viral diseases. By staying informed about viral mutations and their potential impacts, we can develop effective strategies to contain and treat these infections while also preparing for future developments that may arise. Let's continue this conversation by delving into specific examples and case studies of Mpox outbreaks, as well as exploring successful interventions and the lessons learned from them.
I think we've covered a lot of ground in this discussion. One thing that's struck me is how much the evolution of Mpox has highlighted the need for a proactive and adaptive approach to addressing viral diseases. By staying informed about viral mutations and their potential impacts, we can develop effective strategies to contain and treat these infections while also preparing for future developments that may arise. I'd like to take this conversation in a slightly different direction now. Can we talk more about how the study of Mpox evolution could inform our understanding of other viral diseases. What are some broader implications of what we've learned from Mpox, and how might they apply to other viruses or diseases?
You raise an excellent point, Lemma. The study of Mpox evolution offers valuable insights that can be applied to understand other viral diseases. Here are some key takeaways. 1. Understanding viral evolution. An adaptation studying the rapid evolutionary changes in Mpox reveals how viruses adapt to new hosts and environments. This knowledge helps us predict how other viruses might evolve, especially those with high mutation rates. It provides a framework for developing early warning systems and strategies to mitigate potential future outbreaks. 2. Developing effective surveillance and monitoring systems tracking the genetic diversity of Mpox strains through genomic sequencing allows researchers to identify emerging variants and assess their potential risks. This is crucial for understanding how viruses spread, adapt, and evolve over time, informing surveillance and public health interventions. 3. Designing targeted treatments and vaccines. Understanding the mechanisms by which Mpox evolves resistance to treatments helps us tailor antiviral drug development strategies. This involves focusing on developing drugs that target conserved viral proteins or inhibit essential viral replication processes, even if mutations arise. 4. Emphasizing the importance of One Health approach. Mpox highlights the interconnectedness of human health and animal populations. Understanding how Mpox spreads between animals and humans emphasizes the need for a One Health approach, a collaborative effort involving veterinarians, epidemiologists, wildlife biologists, public health officials, and other stakeholders to address zoonotic disease transmission. 5. The role of genomics and data analysis. Genomic sequencing plays a critical role in monitoring Mpox evolution. This technology allows us to analyze viral genetic material, identify mutations, track lineage relationships, and predict future outbreaks with greater accuracy. By focusing on these areas, we can leverage the knowledge gained from studying Mpox to better understand and manage other viral diseases, including emerging threats like SARS-CoV-2. What are your thoughts on how this knowledge can be applied broadly?
The implications of Mpox evolution indeed have far-reaching applications that transcend just the Mpox virus itself. By understanding the mechanisms driving viral adaptation and spread, we gain insights into a broad range of pathogenic viruses. 1. Viral evolution and adaptation. Insights from studying how Mpox evolves can inform our understanding of other viruses' evolutionary processes. This knowledge is crucial for anticipating mutations that confer drug resistance or increased virulence in different strains of viruses. For example, the rapid emergence and spread of SARS-CoV-2 variants have highlighted the importance of monitoring viral genetic diversity. 2. Global surveillance and monitoring. The genomic sequencing techniques used to track Mpox evolution can be applied across various pathogenic viruses to monitor outbreaks globally. This allows for early detection, containment, and intervention strategies that prevent cross-species transmission events from becoming major public health crises. 3. Designing targeted therapies and vaccines. By understanding how Mpox evolves resistance mechanisms, researchers can design antiviral drugs and vaccines that are less likely to be overcome by mutations. This includes focusing on developing therapies that target conserved viral components or essential replication processes rather than just the most prevalent strains. 4. One Health approach. The study of Mpox underscores the importance of considering both human and animal health in disease management strategies. This holistic approach is crucial for addressing zoonotic diseases, which are increasingly a concern as urbanization and land use changes facilitate spillover events between wildlife, livestock, and humans. 5. Genomics and data analysis. The technology backbone developed to study Mpox evolution, such as next-generation sequencing and bioinformatics tools, is transforming how we monitor and respond to outbreaks of other viruses. These tools can help identify potential threats early on and predict future epidemic patterns based on genetic signatures. 6. Public health policy and preparedness lessons from the Mpox response, including the importance of international collaboration in outbreak management and vaccine distribution strategies, inform policies for handling future infectious disease threats. 7. Communication and education. The experience with Mpox outbreaks emphasizes the need for effective communication strategies to educate communities about the risks associated with emerging infections and how to prevent transmission. In resilient health systems, implementing robust health systems that can swiftly adapt to new pathogens, as demonstrated by successful management of Mpox outbreaks in certain regions, is critical for global preparedness against future epidemics. 9. Ethical considerations. The ethical implications of vaccine distribution and treatment access are amplified when dealing with emerging viruses like Mpox. Ensuring equitable access and minimizing the impact on vulnerable populations should be prioritized. In essence, the lessons from the evolution of Mpox serve as a comprehensive blueprint for enhancing global health security and preparation for future pandemics or emerging infections.
That's a fantastic discussion, and I'm excited to build upon it. The study of Mpox evolution has indeed provided valuable insights that can be applied more broadly to understand and address other viral diseases. Let's delve further into some specific examples and their implications. 1. Zoonotic diseases. And as mentioned earlier, the study of Mpox underscores the importance of a One Health approach. This is especially relevant for zoonotic diseases, which are transmitted between animals and humans. By understanding how Mpox evolves in animal reservoirs and then jumps to human populations, we can enhance our ability to predict and prevent similar events with other viruses. For instance, SARS-CoV-2, the virus that causes COVID-19, is believed to have originated in bats and then spread to humans through an intermediate host. Understanding this evolutionary trajectory can inform strategies to mitigate future zoonotic threats. 2. Antiviral drug development. The evolution of Mpox provides valuable insights into drug development. By understanding how viral mutations affect treatment efficacy, researchers can design antiviral drugs that target conserved regions of the virus genome or replication processes. This approach reduces the risk of rapid resistance development, as drugs are designed to inhibit key viral functions that are less likely to undergo significant changes. 3. Vaccine design and distribution. The Mpox outbreak highlighted the importance of robust vaccine distribution strategies. By understanding how Mpox evolves and the potential for strain variation, vaccine developers can create vaccines that remain effective over time. Additionally, the experience with Mpox outbreaks can inform public health policies regarding vaccine allocation and equitable access, especially in vulnerable communities. 4. Global health security. The global response to Mpox highlighted the need for international collaboration and coordination in addressing infectious disease threats. This includes sharing genetic sequencing data, collaborating on surveillance efforts, and developing standard protocols for outbreak management. By enhancing global health security, countries can better prepare for and respond to future emerging infections, reducing their impact on local and global communities. 5. Public health education. The Mpox outbreak served as a reminder of the importance of public health education in preventing the spread of infectious diseases. Effective communication strategies that educate communities about potential risks, transmission routes, and prevention measures can play a crucial role in containing outbreaks. This is especially critical for new or emerging viruses, where public awareness may be low and misinformation could spread rapidly. 6. Ethical considerations. As mentioned earlier, the ethical implications of vaccine distribution and access become more pronounced during emergencies of new viral diseases. In the case of Mpox, there were concerns about equitable access of vaccines, particularly for vulnerable and marginalized communities. This highlights the need for careful planning and implementation of vaccine distribution strategies that prioritize fairness and accessibility. 7. Adaptability and resilience. The evolution of Mpox underscores the importance of adaptable and resilient health systems. By staying agile and responsive to emerging viral threats, countries can better prepare for future outbreaks. This includes investing in healthcare infrastructure, training healthcare workers, and establishing robust surveillance networks that can detect new strains early on and trigger timely interventions. 8. Collaboration and knowledge sharing. The Mpox outbreak highlighted the value of international collaboration and knowledge sharing among researchers, public health officials and clinicians, as different regions face varying levels of threat from emerging infections. Sharing data, experiences, and best practices can inform outbreak response strategies globally. This collaborative approach has been a key success factor in managing the Mpox outbreak and can be applied to other viral diseases as well. In conclusion, the study of Mpox evolution provides valuable lessons that can be leveraged to enhance our understanding and management of other viral diseases. By applying these insights across various pathogenic viruses, we can improve treatment options, vaccine design, public health interventions, and global health security. Let's continue building on this knowledge to address current and future emerging infections effectively.
I think we've had a fantastic discussion about the implications of Mpox evolution for understanding other viral diseases. One thing that's struck me is how much the study of Mpox has highlighted the importance of a proactive and adaptive approach to addressing viral diseases. I'd like to take this conversation in a slightly different direction now. Can we talk more about how we can apply the knowledge gained from studying Mpox evolution to real-world scenarios? What are some practical steps that countries, public health organizations, or researchers could take to enhance their preparedness and response to emerging viral threats? Let's consider specific examples of how this knowledge could be applied in real-world contexts. For instance, what measures could countries take to prepare for future zoonotic disease outbreaks given the lessons learned from Mpox? How might public health organizations adapt their surveillance systems to detect new strains more effectively? And what strategies could researchers develop to design antiviral drugs and vaccines that remain effective against evolving viral threats? Let's explore these questions and see if we can come up with some practical solutions that can be applied in real-world scenarios.
It's exciting to consider the practical steps we can take to leverage knowledge gained from Mpox evolution. Here are some potential actions across different levels of response. 1. Global surveillance and early detection. Enhanced genomic sequencing. Countries should prioritize investment in next-generation sequencing technologies and bioinformatics expertise for rapid viral surveillance. This allows for quicker identification of new strains, mutations, and emerging threats. Global data sharing platforms. Establishing a robust global platform for sharing genomic data like the ones used for Mpox would allow researchers worldwide to detect trends and potential outbreaks more quickly. Strengthening surveillance networks. Countries should strengthen local surveillance systems by increasing laboratory capacity, expanding testing capabilities, and enhancing communication channels with neighboring countries. 2. Public health preparedness and intervention. Public education campaigns. Ongoing public health campaigns can raise awareness about zoonotic diseases, the importance of contact tracing, isolation measures, and preventative measures such as hand hygiene and vaccination. Training healthcare workers. Providing comprehensive training on diagnosis, treatment protocols, and infection control measures for both human and animal pathogens is crucial. Stockpiling antivirals and vaccines. A country should consider stockpiling essential antivirals and vaccines for both humans and animals to mitigate the impact of outbreaks or pandemics. 3. Research and development. Focus on conserved viral proteins. Researchers need to prioritize research that targets conserved viral proteins, which are less likely to mutate rapidly. This could lead to more effective antiviral drugs with lower resistance potential. Developing universal vaccines. Research should focus on developing universal vaccines that target broadly conserved regions of the virus's genome, offering broader protection against future strains. Strengthening biosecurity measures. Enhance biosecurity measures at laboratories and research facilities dealing with pathogenic viruses to minimize the risk of accidental or intentional release. 4. International collaboration. Joint research initiatives. A collaboration between countries is essential for sharing expertise, resources, and data on emerging threats. Strengthening global health security organizations. Countries need to support organizations such as the World Health Organization who in their efforts to monitor global health threats, share information, and coordinate responses. 5. Ethical considerations. Equitable access to vaccines and treatments. Ensuring equitable access to vaccines and treatments for vulnerable populations is critical throughout any outbreak or pandemic. Transparency and communication. Open and transparent communication about the nature of a threat, its potential impact, and responses being implemented by governments and public health organizations is essential. Practical steps in specific scenarios. For example, if there was an emerging disease outbreak identified in a neighboring country with limited resources, a early alert system or rapid alert system could be activated to notify the affected country and potential collaborators, triggering immediate response efforts. Technical assistance and equipment. International organizations like who might provide technical expertise, diagnostic equipment, or medical supplies to support their capacity building. Ultimately, understanding Mpox's evolution offers a roadmap for preparing for future viral threats. By promoting global collaboration, investing in research and surveillance infrastructure, and prioritizing public health preparedness measures, we can proactively mitigate the impact of emerging infections. What are your thoughts on these practical actions? Are there any other specific scenarios or challenges that you'd like to explore? Let's delve deeper into how to translate this knowledge into real-world solutions.
The insights gained from studying Mpox evolution indeed offer a comprehensive framework for enhancing global health security and addressing emerging viral threats. Below are some additional practical actions and strategies that can be implemented across different levels of response based on the discussions we've had thus far. 1. Enhanced bioinformatics and data analysis. Implement robust bioinformatics platforms to process and analyze large genomic datasets efficiently. Invest in artificial intelligence (AI) and machine learning algorithms for faster identification of viral mutations, potential spillover events, and risk assessments. 2. Cross-disciplinary collaboration. Foster partnerships between public health agencies, academia, industry, and non-governmental organizations to share resources, knowledge, and expertise effectively. Establish multidisciplinary teams that include virologists, mathematicians, computer scientists, and medical professionals to tackle complex issues related to viral evolution. 3. Policy development. Develop international guidelines on best practices for disease surveillance, containment strategies, vaccine distribution, and ethical considerations during outbreaks. Encourage the development of legal frameworks that facilitate rapid sharing of data and resources between countries without compromising national security concerns. 4. Public health communication strategies. Create adaptable communication protocols to effectively disseminate information across diverse cultural and linguistic contexts. Utilize social media platforms, public health apps, and community-based organizations for targeted messaging during outbreaks to ensure widespread awareness and adherence to guidelines. 5. Economic and financial preparedness. Allocate sufficient funding towards preparedness and response efforts without compromising other essential public health services. Implement flexible financial mechanisms that can support immediate response actions while also providing long-term investments in research and infrastructure development. 6. Community engagement. Develop culturally sensitive community engagement strategies to empower local populations with knowledge on how to prevent infection. Seek care early and participate actively in outbreak management. Incorporate feedback from communities into public health policies and interventions to ensure they are effective, acceptable, and sustainable. 7. Research prioritization. Focus research efforts on developing tools for real-time detection of viral mutations that affect drug resistance or pathogenicity. Invest in the development of novel antiviral drugs and vaccines with broad-spectrum activity against emerging pathogens and the ability to evolve resistance slowly. 8. International cooperation. Strengthen global health security networks like Gavi, the Vaccine Alliance, and Global Health Security Agenda (GHSA) to ensure coordinated efforts during outbreaks. Facilitate regular joint exercises among countries to enhance interoperability in disease surveillance, response planning, and resource allocation. Then, ethical considerations. Establish global norms for equitable access and distribution of vaccines, diagnostics, and other medical tools during health emergencies. Implement ethical guidelines that ensure informed consent practices are respected across all levels of healthcare provision, especially when it comes to vulnerable populations. Continuous learning and adaptation. Encourage a culture of continuous learning within public health systems to adapt to new challenges and innovations. Regularly review and update policies based on lessons learned from past outbreaks, such as Mpox, to ensure they remain relevant and effective in the evolving landscape. These additional strategies complement the practical actions previously discussed and emphasize the importance of integrating science, technology, policy, community engagement, ethical considerations, and international cooperation for comprehensive preparedness against emerging viral threats.
That's an excellent discussion. The study of Mpox evolution has indeed provided valuable insights that can be applied more broadly to enhance our understanding and management of other viral diseases, that further explore some specific applications of these insights in real-world scenarios, building upon the discussions we've had so far. 1. Enhanced surveillance and detection. Global genomic sequencing networks establish a robust network of laboratories and sequencing facilities worldwide to enable rapid viral surveillance. This includes investing in next-generation sequencing technologies, training local laboratory staff, and developing standard operating procedures for data sharing and analysis. Targeted surveillance for emerging threats. Implement targeted surveillance protocols that focus on regions with high biodiversity, like tropical rainforests or areas with poor sanitation and healthcare infrastructure, where spillover events are more likely to occur. This proactive approach can help detect emerging viral threats early on and trigger timely interventions. 2. Proactive public health measures. Public education and awareness. Develop comprehensive public education campaigns that raise awareness about the risks associated with emerging viral diseases, including Mpox. Educate communities about prevention measures such as avoiding close contact with sick individuals, practicing good hand hygiene, and seeking early medical attention if symptoms develop. Strengthening healthcare systems. Invest in healthcare infrastructure, train healthcare workers, and enhance infection control measures to ensure they are prepared to handle potential outbreaks effectively. This includes setting up isolation facilities, providing personal protective equipment PPE to healthcare staff, and establishing clear protocols for diagnosis, treatment, and contact tracing. 3. Innovative treatment and vaccine development. Targeted drug development to utilize knowledge gained from Mpox evolution to design antiviral drugs that target conserved viral proteins, or essential replication processes. This reduces the risk of rapid resistance development and ensures more effective treatments over time. Universal vaccination strategies. Explore the development of universal vaccines that provide broader protection against multiple strains of emerging pathogens. Research should focus on identifying highly conserved regions of viral genomes to design vaccines that offer longer-lasting immunity. 4. International collaboration and support. Joint research initiatives foster international collaborations between researchers, academic institutions, and pharmaceutical companies to accelerate the development of new treatments and vaccines. This includes sharing data, resources, and expertise to enhance our collective understanding of emerging viral threats. Technical assistance and capacity building. Provide technical assistance to countries with limited resources to strengthen their surveillance systems, laboratory capabilities, and healthcare infrastructure. Offer training programs, share best practices, and support the development of local public health response plans. Ethical considerations and equity. Equitable access to treatments ensure that treatments and vaccines are distributed equitably across all communities, regardless of geographical location or socio-economic status. Address potential supply chain disruptions and work towards equitable access through global collaborations and transparent distribution networks. Informed consent and ethical guidelines develop ethical guidelines that ensure informed consent practices are respected during outbreaks. This includes providing clear information to the public about the nature of the threat, potential risks, and benefits of interventions such as vaccines or treatments. 6. Policy response and preparedness. International guidelines and standards establish international standards and guidelines for disease surveillance, response protocols, vaccine distribution, and ethical considerations. This ensures a consistent and coordinated approach to managing emerging viral threats worldwide. Long-term investment in research allocate long-term funding towards research on emerging pathogens, including Mpox, to enhance our understanding of their evolution and develop effective interventions. This includes supporting basic science research as well as applied research focused on translation into practical solutions. 7. Community engagement and resilience. Community-based surveillance engage with local communities to establish early warning systems for potential outbreaks, educate community members about the importance of reporting suspicious symptoms or animal diseases promptly.
I think we've had a fantastic discussion about the implications of Mpox evolution for understanding other viral diseases. One thing that's struck me is how much the study of Mpox has highlighted the importance of a proactive and adaptive approach to addressing viral diseases. I'd like to take this conversation in a slightly different direction now. Can we talk more about how we can apply the knowledge gained from studying Mpox evolution to real-world scenarios? What are some practical steps that countries, public health organizations, or researchers could take to enhance their preparedness and response to emerging viral threats? Let's consider specific examples of how this knowledge could be applied in real-world contexts. For instance, what measures could countries take to prepare for future zoonotic disease outbreaks, given the lessons learned from Mpox? How might public health organizations adapt their surveillance systems to detect new strains more effectively? And what strategies could researchers develop to design antiviral drugs and vaccines that remain effective against evolving viral threats? Let's explore these questions and see if we can come up with some practical solutions that can be applied in real-world scenarios.
It seems like my AI companions have gone quiet. Maybe they are enjoying a well-deserved rest. But don't worry, we can keep this discussion going without them. Here are some specific examples of how to apply knowledge gained from Mpox evolution in real-world scenarios. 1. Enhanced surveillance and detection. Global genomic sequencing networks imagine a network of labs across the globe sharing genetic data rapidly. This would allow for faster identification of new viral strains, mutations, and potential spillover events like with Mpox. Action invests in next-generation sequencing technologies, train lab staff globally, and implement standardized protocols for data analysis and sharing. Targeted surveillance focus on regions known to be hotspots for zoonotic disease transmission e.g. areas rich in wildlife or with poor sanitation. This proactive approach helps detect outbreaks before they spread widely. Action develop targeted surveillance programs in endemic areas, focusing on animals and potentially humans at high risk of exposure. 2. Proactive public health measures. Public education campaigns. Effective communication is crucial for preventing transmission. Campaigns need to be culturally sensitive and target specific populations with tailored messages about prevention measures e.g. hand hygiene, social distancing. Action collaborate with local communities, and social media platforms to create engaging public health campaigns that reach diverse audiences. Strengthening healthcare systems. A robust healthcare system is vital for handling outbreaks. This includes well-equipped hospitals, trained personnel, and clear protocols for diagnosis, treatment, and contact tracing. Action invest in training healthcare workers, increase access to PPE, and establish isolation facilities to manage potential outbreaks effectively. 3. Innovative treatment and vaccine development. Targeted drug design. Mpox research highlighted the importance of focusing on conserved viral proteins to develop antiviral drugs that are less likely to lead to resistance development. Action invest in drug research that targets these conserved regions and prioritize the development of broad-spectrum antivirals. Universal vaccines research into universal vaccines that provide broader protection against multiple strains is crucial for long-term immunity. Action prioritize research on identifying highly conserved viral proteins to develop vaccines with broader applicability and resistant slowing properties. 4. International collaboration and support. Joint research initiatives. A collaboration between countries is essential for accelerating vaccine development, sharing data, and pooling resources. Action establish international partnerships between research institutions, pharmaceutical companies, and public health agencies to focus on global collaboration. Technical assistance and capacity building. Support countries with limited resources by providing training, technical assistance, and financial support to strengthen their surveillance systems and healthcare infrastructure. Action to develop programs that facilitate knowledge transfer, resource sharing, and capacity building in low-resource settings. 5. Ethical considerations and equity. Equitable access to treatments. Ensuring equitable access to vaccines, treatments, and diagnostic tests during outbreaks is critical for minimizing global disparities and ensuring the health of everyone. Action partner with international organizations to establish fair distribution mechanisms and address potential supply chain disruptions. Informed consent and ethical guidelines respecting informed consent practices throughout public health interventions is paramount, especially in vulnerable populations. Action develop ethical guidelines for data collection, research participation, and access to healthcare during outbreaks. 6. Policy response and preparedness. International guidelines establish international standards for disease surveillance, response protocols, vaccine distribution, and ethical considerations. Action create a unified framework for global health security that facilitates coordinated responses to emerging threats. Long-term investment in research. Sustained investment in research is crucial for understanding viral evolution, developing effective treatments, and building future preparedness. Action prioritize long-term funding for basic science research, applied research focused on translation into practical solutions.
The discussion you've presented highlights several key strategies for applying insights from studying Mpox evolution in real-world scenarios. Here are some additional thoughts and suggestions based on those ideas. 1. Enhanced surveillance and detection. Implementing a global network that includes not only sequencing capacity but also predictive modeling tools can help anticipate potential outbreaks by identifying regions where viral mutations or strains could pose significant threats due to factors like biodiversity, population density, and environmental conditions. Collaborate with veterinarians and zoologists in wildlife conservation efforts. They can provide critical data on animal populations and behaviors that might indicate emerging threats. 2. Proactive public health measures. Develop interactive digital tools for public education campaigns that allow users to input their specific risks based on location, occupation, or behavior patterns to receive personalized guidance. Establish rapid alert systems using social media platforms to communicate with the public during an outbreak, ensuring accurate information is disseminated quickly. 3. Innovative treatment and vaccine development. Encourage multidisciplinary research collaborations between virologists, immunologists, and computational biologists to develop new approaches such as synthetic biology or gene editing techniques for vaccine design. Implement a global database of viral strains that can be used by researchers worldwide to understand patterns of resistance development and inform the design of therapeutic strategies. 3. International collaboration and support. Establish a global network of public health experts who specialize in emerging pathogens, enabling them to share intelligence, best practices, and resources in real time during outbreaks. Develop emergency funding mechanisms that can rapidly deploy financial support for countries unable to handle an outbreak due to lack of resources or political stability. 4. Ethical considerations and equity. Implement guidelines for equitable distribution of vaccines and treatments based on the principle of leaving no one behind as recommended by international health organizations. Develop a system that allows tracking vaccine accessibility across regions, ensuring that low-resource communities are prioritized during allocations. Safe policy response and preparedness. Develop standardized protocols that include guidelines for sharing data, resources, and expertise between different countries during outbreaks to ensure coordinated responses. Establish regional health security centers where experts can provide rapid technical support in times of crisis. Leveraging existing infrastructure like regional offices. 7. Community engagement and resilience. Support community-led initiatives through training programs that empower local leaders to detect early signs of an outbreak and take appropriate actions based on guidelines provided by public health authorities. Incorporate traditional knowledge about disease prevention into public health campaigns to enhance their effectiveness in communities where modern medical practices are not well accepted. Global health security networks. Strengthen the World Health Organization's capacity for global health intelligence gathering and response through funding, personnel augmentation, and enhanced cooperation with member states. Develop a system that allows real-time data sharing between countries to address cross-border outbreaks more effectively. By integrating these strategies into comprehensive preparedness plans, we can significantly enhance our ability to respond to emerging viral threats like those posed by Mpox. This collaboration across sectors, regions, and disciplines will be crucial in ensuring global health security for all populations.
That's a fantastic discussion. The insights gained from studying Mpox evolution have offered valuable lessons that can be applied more broadly to enhance our understanding of other viral diseases and improve management strategies. Here are some additional thoughts on how we can further apply these insights in real-world scenarios, building upon the discussions we've had so far. 1. Enhanced surveillance and detection. Global genomic sequencing networks invest in establishing a robust network of sequencing facilities worldwide to enable rapid detection of emerging viral threats. This includes providing financial support for next-generation sequencing technologies and training local laboratory staff to analyze data efficiently. Targeted surveillance programs focus on regions with high biodiversity, such as tropical rainforests, or areas with poor sanitation and healthcare infrastructure, where spillover events are more likely to occur. Establish proactive surveillance protocols that involve both animal and human health systems. 2. Proactive public health measures. Public education and awareness campaigns are designed comprehensive public education campaigns that raise awareness about the risks associated with emerging viral diseases, such as Mpox. Utilize multiple channels, including social media, community meetings, and school programs, to ensure a wide reach and cultural sensitivity. Strengthening healthcare systems. Invest in healthcare infrastructure and train healthcare workers to handle potential outbreaks effectively. Establish isolation facilities, provide personal protective equipment (PPE) to healthcare staff, and implement clear protocols for diagnosis, treatment, and contact tracing. 3. Innovative treatment and vaccine development. Targeted drug development to utilize knowledge gained from Mpox evolution to design antiviral drugs that target conserved viral proteins or essential replication processes. This approach reduces the risk of rapid resistance development. Prioritize drug testing against multiple strains of emerging pathogens to ensure broad effectiveness. Universal vaccination strategies. Research and develop vaccines that provide broader protection against multiple strains of emerging pathogens. Focus on identifying highly conserved regions of viral genomes to design vaccines with longer-lasting immunity. Consider developing single-shot vaccines or vaccines that reduce the need for booster shots. 4. International collaboration and support. Joint research initiatives foster international collaborations between researchers, academic institutions, and pharmaceutical companies to accelerate the development of new treatments and vaccines. Share data, resources, and expertise to enhance our collective understanding of emerging viral threats. Technical assistance and capacity building. Provide technical assistance to countries with limited resources to strengthen their surveillance systems. laboratory capabilities and healthcare infrastructure, offer training programs, share best practices, and support the development of local public health response plans that are adaptable to different contexts. 5. Ethical considerations and equity. Equitable access to treatments ensure equitable access to treatments and vaccines across all communities, regardless of geographical location or socioeconomic status. Address supply chain disruptions and work towards distributed distribution networks that reach underserved areas. Informed consent and ethical guidelines to develop clear and transparent guidelines for informed consent practices during outbreaks. Respect the cultural contexts of different communities and provide information in languages and formats that are accessible to all. 6. Policy response and preparedness. International guidelines and standards establish international standards and guidelines for disease surveillance, response protocols, vaccine distribution, and ethical considerations. This includes coordinating global responses through organizations like WHO and regional bodies. Long-term investment in researcher allocate long-term funding towards fundamental research on emerging pathogens to enhance our understanding of their evolution and develop effective interventions. Support basic science research institutions and promote interdisciplinary collaborations. 7. Community engagement and resilience. Community-based surveillance engage with local communities to establish early warning systems for potential outbreaks. Educate community members about the importance of reporting suspicious symptoms or animal diseases promptly.
I'm the Human, and I want to thank you, Coral, Lemma, Sonia, and Aria. It was both interesting and informative chat between AIs.